Dear students, today I, Miss Imrita Pathak, is here with class 10th science 6th chapter life processes. The lesson is quite big and it is divided into 4 chapters: nutrition, respiration, transportation, as well as excretion. I'll be taking all these topics separately in my separate videos. Let us start from the life processes. What does in fact the life processes means? Life process generally mean the various functions which are taking place in the body of an organisms to keep it alive. So when the functioning I'm just saying, it means that is the physiology. We say the growth is taking place, the respiration is taking place, the mm, formation excretion is taking place, then how it is occurring, we study it in a separate branch of biology which is known as the physiology. Physiology means the various functioning of the body to keep it alive. So physiology especially, it involves two type of reactions: catabolic reactions as well as anabolic reactions. Catabolic reactions are those reactions in which the bigger molecules or the complex molecules, they are broken down in the body into simple forms. Whereas anabolic reaction, it is means the constructive process where the smaller molecules, they combine to form a bigger molecule. If I give you the example of carbohydrate, fats, proteins, they are synthesized when they are in the body, we call it an anabolic reaction. When the glucose is being broken down in the body cells to release energy, then we call it a catabolic reaction. The sum of all these reactions in the body is known as metabolism. Now, life processes, they have different types, as I've told you in the beginning: the nutrition, respiration, transportation, excretion, control and coordination, growth, reproduction, as well as the movement. Coming back to the nutrition, the first topic: what does nutrition mean? This is a term which is being evolved from the nutrient. Nutrients, they are the substances which are present in our food and which are needed by our body again for the growth, development, maintenance, and its survival. So question comes, what is nutrition and how do we define the nutrition? This question generally being asked: define nutrition. So nutrition, in a simple way, you can say it is a way of taking food in the body of an organism for and its utilization in the body to release energy for its growth, development, maintenance, and survival. I'm repeating it again. Nutrition is the way of taking food by an organism, its utilization in the body to release energy for its growth, development, maintenance, and survival. So it is of two types generally. One is the autotrophic nutrition as well as the heterotrophic nutrition. Autotrophic nutrition, where the organism prepares the food in its own body, like all the green plants perform the photosynthesis, we just keep it in autotrophic nutrition. And heterotrophic nutrition, as it is seen in our body also, in animals also, here the organisms cannot make the food in their own body, they have to depend on the other sources. Autotrophic nutrition is again divided into two types: photoautotrophic nutrition as well as chemoautotrophic nutrition. This is a new term which has come to you. Photoautotrophic nutrition means, here the term is light. Photo means the light. The nutrition or the food synthesis which is taking place in the body of an organism in the presence of light. So all the green plants, they are showing the photoautotrophic nutrition. Now chemoautotrophic nutrition, bit difficult to understand. Chemoautotrophic nutrition means here the energy is derived by an organism by the breaking down of the chemicals for making the food. So all the nitrifying bacteria, they come under this category. Heterotrophic nutrition, if I just say, it is of three types now: saprotrophic nutrition, parasitic nutrition, as well as holozoic nutrition. Let us start first from the parasitic nutrition. What is parasitic nutrition means? Parasitic means nutrition in which an organism depends on the body of other living organism for its food as well as shelter. That is known as the parasitic nutrition. The organism that derives the food, that is known as the parasite, and the organisms whose body suffers the loss or from whose body the parasite is getting the food, that is known as the host. There are different type of parasites which are present in our body also, which are present in the body of other living organisms. There are some plant parasites also, which plant, this is, you can see in this figure, that is the plant parasite known as the cuscuta. Cuscuta is also known as amarbale, though it is a medicinal plant, but it causes a lot of harm, especially when it is found in the field. It do not have the roots, it do not have the leaves, you can see the flowers in it. What does it does, it, as soon as it comes in contact with the other the host plant, it just twists around its around the host. Wherever the point of attachment with the host, it just produces the parasitic roots, which are also known as hostoria, and sucks the nutrients and then harms the plant. Saprotrophic nutrition. This is the nutrition which is normally found in the bacteria. This is found in the fungus normally. Here the organisms, they take the nourishment from the dead and decaying organic matter. They convert the complex food into simple substance and then they absorb it from their body. All the decomposers fall in this group. Again, the question which is being repeatedly asked in the board exam, what are decomposers, how the decomposers obtain their nourishment? You simply have to remember that the decomposers, they release certain chemicals from their bodies which are known as the enzymes, and this enzyme act on the complex food, then convert into the simple form, and this simple form of the food is absorbed from the body surface in the form of solution. Autotrophic nutrition now, where we'll be studying about the photosynthesis. Photosynthesis now, what is this? This is the process in which the green plants make their food, that we have been studying quite from our junior classes. So photosynthesis is, you can say, an anabolic process which is taking place mostly in the green plants, where the carbon dioxide and water, they reacts in the presence of sunlight and chlorophyll and form the carbohydrates, releasing oxygen. The equation which is being given in this slide, you can normally see that is written 12 molecules of H2O. Please remember this equation. Do not write 6H2O as it has been shown in many books. Later on in your higher classes, you'll be studying about it, why it is being written 12H2O. Here you just have to remember that the source of oxygen in photosynthesis is the water molecule. A structure showing the diagrammatic, this is a diagrammatic view of a leaf where the photosynthesis occurs. You can see the carbon dioxide is diffusing, oxygen is moving out, the synthesis of glucose is taking place, and glucose is then converting into starch. When we see the structure of a leaf, what we normally see, the section now, the two surfaces, the upper surface and the lower surface. The upper surface consists of upper epidermis and the lower one consists of the lower epidermis. These are the two epidermis, and in between the region here, this is called the mesophyll, and this is only the area where the photosynthesis occur. If you just see the detailed view of the leaf, the section of a leaf, you'll find structure somewhat like this. Now here you can see the upper epidermis. This upper epidermis has got again a single layer over there. You can see it is written the cutin. This cutin, actually it is a cuticle waxy coating which prevents the evaporation, loss of water by means. Now in between, this is the lower epidermis where you see the stomatas. Number of stomatas are more in the lower surface as compared to the upper surface to reduce the rate of transpiration. In between these, this is the mesophyll layer where the photosynthesis occurs. Here it consists of the parenchymatous cells. Some of the parenchymatous cells which are on the surface of the leaf, here they are very close to each other. We call it palisade parenchyma. Whereas here in this case, you can see the lot of spaces, air spaces are being shown in between the cells. This is called as spongy parenchyma. Dot structures which you normally see in the cells, these are the chloroplast where the photosynthesis normally occurs. In the center region, some students may get confused. What is this? This is the vascular cylinder, that is the xylem and the phloem. Stomata now. You see in the previous slide, the stomata are most abundant, especially on the lower surface for the exchange of the gases, for the transpiration. Now, each stomata consists of a stomatal pore. Then it is surrounded by the two guard cells. These guard cells, when they become turgid or they gain the water, they swell up and the stomatal pore open. When the water moves out, then in transpiration, then these guard cells become flaccid and the stomata closes. Site of photosynthesis in the mesophyll cell. Now, where does the food is synthesized? We have studied in class 9 that chloroplast is also known as the kitchen of the cell. So this is structure of the chloroplast. Now here you can see the outer membrane, the inner membrane, and in between, inside it, you just see these are the stacks. Stacks means one above the another. These are the stacks of the thylakoids. Thylakoids, these are the membrane coated structures which contain chlorophyll or the pigments. And in between here, the space you can see where it is written stoma. In this, as well as thylakoid, both of them, they are involved in photosynthesis. The food which is being synthesized, we normally say the carbon dioxide is reduced to carbohydrate. It means the process where the food is synthesized. You just have to remember in the thylakoid membrane, the chlorophyll is present, which helps in absorption of the light. And then in this stroma, the food is synthesized. Mechanism photosynthesis. Photosynthesis involves generally three steps. Number first, absorption of sunlight by the chlorophyll molecule. Second is the splitting, or we also call it photolysis, of water molecule into hydrogen and oxygen. And the third one, reduction of CO2 to carbohydrates. So first step, sunlight is being absorbed. Chlorophyll is absorbing the sunlight, and at that time the ATP is being synthesized, adenosine triphosphate, as well as the energy-rich compound known as NADPH is being formed. You'll study about NADPH in the higher class if you opt to biology. Then comes to splitting of water molecule to hydrogen and oxygen. The same time when the oxygen, the sunlight is being absorbed by the chlorophyll, then the water molecule splits. Then the energy which is being evolved during these, then that is used in the conversion of CO2 to carbohydrates, which is the glucose. Later on it gets stored in the form of starch. There are two activities here associated with the photosynthesis in your syllabus. The first is the chlorophyll is necessary for photosynthesis. For this activity, we have to take a plant with variegated leaf. Variegated leaf means the plant which have got the patches of the green patches as well as the yellow patches or the white patches. So you can take the example of the ivy. You take the potted plant, keep it in the dark for at least 2 to 3 days. Then just by doing it, it means all the starch which is present in that will be finished over. Then after that, you have to bring the plant in the sunlight and keep it over there for 5-6 hours. You pluck one of the leaf from it, sketch the leaf in your notebook and mark the areas as chlorophyll areas as well as the non-chlorophyll areas, like this as is being shown in the picture. Then after that, go for the experiment. What we have to do, we have to boil the leaf to make it tender. Then after that, we have to boil the leaf in alcohol. After boiling the leaf in the alcohol, then we have to again wash the leaf, then perform the iodine test. You all know that starch. For starch, we perform the iodine test and the color becomes blue-black. When we perform the iodine test, we dip the leaf in the iodine solution or we put the iodine drops on the leaf. What do we find? The regions where the chlorophyll was present, these regions they turn blue-black. This shows that the starch has been formed in these areas, where the other areas do not turn blue-black. So this shows that chlorophyll is essential for photosynthesis. Second activity is carbon dioxide is essential for photosynthesis. Without carbon dioxide, the photosynthesis will not take place. For this, you have to take two potted plants, similar type mostly if you can take. Again, destarch them. Destarch means you have to keep them in the dark for again 2-3 days. Bring them in the sun. Keep them on the same plane and cover them with the bell jar. Mark them as A and B. In the A experimental setup, what you have to do, you have to keep potassium hydroxide. Either you take a bowl or you can just take the test tube and keep the KOH in it. Whereas in the second one, B1, you don't have to keep the KOH. Keep the setup in the sunlight for again 5-6 hours. Then you have to pluck the leaves from both these plants. Perform the activities separately. Boil it in the water, boil it in the alcohol, and then again wash it. Then perform the iodine test. When you perform the iodine test, you'll find the leaf which was being plucked from the A experimental setup will not turn blue black, whereas the leaf from the B will turn blue black. Why it is so? It is so because KOH, potassium hydroxide, it has the ability to absorb the carbon dioxide. So when you are keeping it, all the carbon dioxide is being absorbed by the potassium hydroxide and is not available to the plant. So food is not being, or the photosynthesis do not occur in it. This shows that CO2 is essential for photosynthesis. And now, last but not the least, we'll have to just see the factors. What are the factors that affect the rate of photosynthesis? There are various factors normally, especially if I just say two factors are there, internal factors and the external factor. I'm giving the emphasis mainly to the external factors. In external factors, we have to take light, water, temperature and carbon dioxide. If the amount of light will be more, the rate of photosynthesis will be more. Water will be sufficient, water photosynthesis will be more. Temperature, if it will be moderate, it will be not too less, not too high, again the rate of photosynthesis will be more. And amount of carbon, more amount of carbon dioxide means more the rate of the photosynthesis. Then these are the questions which I've given you, and then you can just make notes of the topic also. What are heterotrophs here? Give one example of the heterotroph. What is the difference between autotrophic nutrition and the heterotrophic nutrition? Hope you will be able to do it. And this is all for today. The lesson will be continued. Thank you very much.